Hello and welcome to EFB103. This is Chapter 3, Carbon and the Molecular Diversity of Life, Part 1. So the main topics for this chapter are listed below. Carbon are going to be a central theme, the four main macromolecules and their structure, and then general functions of these macromolecules, which will blend into them as we talk about them. So let's get started looking at carbon. Here are some examples of our important macromolecules that make up living things. And you'll see if you look at the chemical structure here, a central component to all of these is carbon. So carbon is this very versatile element that we can use as our skeleton for our organic compounds in living things. Why is it so versatile? Well, think back to chemical bonding and those valence electrons in that outer shell of our atoms. If we look at some examples here, hydrogen, oxygen, nitrogen, look at the number of valence electrons carbon has. It has 4. What does that mean? That means it can make 4 covalent bonds. So it acts as almost like a hub, I like to think of it like that, to add other things onto. And that can be true for nitrogen as well, but you can get 3 things on nitrogen, 3 covalent bonds, 2 on oxygen. And that can kind of act as a bridge between 2 things, but carbon can act as that central focal point of that skeleton. All right, so what does it look like when we start adding things to a carbon atom? Well, we can take the example of methane here, the top row of our table. And if we were to just draw this on a piece of paper, we'd put a carbon in the middle and 4 hydrogens coming out north, south, east, and west. What's really happening, though, in 3 dimensions is that those hydrogens will radiate out from that central carbon really equidistant from each other, and they can't get far enough away from each other if they were just to stick out in a plane like we see in this structural formula. But really, they form a tetrahedral shape. So these ones kind of angle down, and that way they're all the same distance from each other. You see the same pattern when you, say, take 2 carbons here in the middle. Carbon can bond to carbon, and then we just throw hydrogens on the rest of those electrons. And you see each carbon still has that tetrahedral shape. Those things can rotate because it's a single bond, so that could really spin around. And that is in contrast with something like ethene here, where the 2 carbons in the middle have a double bond. That really locks them in place. So this prevents that free rotation in that carbon-carbon bond, and it creates really more of a planar structure. So as we've drawn it here, say on our piece of paper in 2 dimensions, the 3-dimensional model looks very similar to that. Those hydrogens radiate out in this plane. So there's other ways to visualize this, of course. Taking our electron mapping type diagram here, where our carbons and our shared electrons between them, and then the shared electrons with the hydrogen groups here. And so this is a hydrocarbon if we have carbon and hydrogen. So we see that theme a lot in biological molecules. But carbon can bind with more things than just hydrogen, of course. And here's an example of carbon dioxide. So carbon in the middle and then 2 oxygens on either side. And notice these are double bonded. There's 2 pairs of electrons shared in both oxygen molecules with this carbon. And so these bonds themselves, carbon, oxygen, those are polar bonds. But this came up before. Carbon dioxide, it ends up being a nonpolar molecule because it has this symmetry where the oxygens are directly opposing one another. All right, so let's start adding more things on here. And so a couple of important points as we start to make more complex molecules is, well, how long is that chain of carbons? If we add a carbon, we have something that's 1 carbon longer, and we can keep adding those and we get different compounds. Big surprise. But it gets more interesting when we have 4 or more carbons, because given a chemical formula, so in this case, C, 1, 2, 3, 4, and H10, there's 10 hydrogens here, you can end up with the same, use that same chemical formula, but look, you have a different compound because the carbons are bonded differently. We've arranged them differently. Where the double bond is might matter if we got a compound with a double bond. So here is a compound, 4-carbon compound, and the double bond is over here, versus this one, it's between the two central carbons. That creates a different compound, 1-butene and 2-butene. Carbon can form these rings. So here in this cyclohexane example, the carbons all have single bonds between them. In the benzene example, every other carbon pair has a double bond between them. So where those bonds occur, where the double bonds occur, how they're arranged, all of a sudden you can start to see where this complexity can arise. So when we have these different structures that come out of the same chemical formula, like here C5H12, we can make pentane or 2-methylbutane, and we've just arranged those same atoms in a different way. These are called isomers. So something has the same formula, but we've created a different compound structurally. They're isomers. And of course, if we add more and more carbons, there's just more and more possible isomers that we could create from them by putting things together in different ways. Another important type of isomer are the cis-trans isomers. So in this case, as demonstrated here in this figure nicely, you have a carbon-carbon double bond, and this locks those carbons in place so they don't rotate freely in that planar structure. And because of that, you could have these other groups binding here, whatever they happen to be. They're just annotated by X's in this example. In this cis isomer, these two X groups of interest are on the same side, whereas the trans isomer, notice they're not on the same side. They're on opposite sides of this double bond. And that's going to matter, right? Because this is going to give you a different structure than this. Again, even though very similar, or identical if you write out the chemical structure, or the chemical formula rather, but the one difference of the orientation of these groups can give you a different acting compound. Another kind of isomer is called an enantiomer. And these are mirror image isomers. So this idea that you can have left or right-handed versions of the same molecule. And you know, the hand example is a really good one. You know, you take your hands, face them towards you, palms towards you, and then overlay them. They don't overlap perfectly. Your thumbs are going to stick out either side. If you turn them and face each other, so they're mirrors of each other, you can put them together, but not if you try to overlay them. And that's the same idea with these compounds. So in this example here, we have a left and right compounds. And if we were just to look at the chemical formula, it's going to be the same. It looks pretty much the same structurally, but we couldn't overlap them. They're oriented, they're mirror images of one another. Let's start putting together some of our basic knowledge now into bigger things. So one of the common themes or patterns or elements that you'll see when you look at structures of organic compounds in living things are these functional groups. These are common groups of atoms that you see that make up these compounds. So an example at the bottom here are estradiol and testosterone. Highlighted in blue are some of these functional groups. In this particular case, these are functional groups that are important because they participate in a chemical reaction. But really a functional group is something that helps give that compound its properties. And so what are some of those functional groups that we see in living things? Well, here they are. Here's the list of them, and we'll look more closely at them. But there are 7 kind of key functional groups. And one of the questions we might have about all of these is fundamentally, are they polar or non-polar? Because that's going to determine whether that compound, or at least that part of that compound, interacts with water or does not. So that's really important, and why we were looking at this earlier. How do we know whether a bond is polar or non-polar, and therefore that functional group polar or non-polar? We go back to our relative electronegativities, and we can determine whether that bond is polar and therefore whether that functional group is polar. So let's take the example of alcohol first. So alcohol is a hydroxyl group, just this OH. And if we go back to our table, oxygen and hydrogen, the difference is 1.4 in electronegativity. That is much greater than the 0.5, so this would be a polar bond. So this is a polar functional group. Now, on the end of alcohol here, that's pretty much what makes alcohol able to interact with water, because if you look at the rest of alcohol, it's just this carbon hydrogen. And if we go back to our electronegativity, carbon and hydrogen, there's not enough electronegativity difference here to make this a polar bond. Nonpolar, doesn't interact well with water. The next one here, carbonyl group, that's C double bond O. Carbon oxygen bonds are polar, so we see that here with carboxyl group 2. So oxygen double bond and an OH group. This hydrogen can actually dissociate in carboxyl, and this makes the solution more acidic. So this is also known as a carboxylic acid group. Amino group, nitrogen and hydrogen. What's the difference between those two? Let's go look. Nitrogen, hydrogen, yes, bigger difference than 0.5, looking good. Let's keep it rolling. Sulfhydryl group, sulfur and hydrogen. Is this a polar or non-polar bond? I'll go back and you can answer that. All right, so you've had a look and you've answered the question. This is a moderately polar bond, that's SH bond. If we go back to our table, that rule of thumb, it's really borderline, right? And so that's where a rule of thumb is just that. It's a rule of thumb. It's really close on this one, but that SH bond is moderately polar. How about phosphorus and oxygen? Very polar. We'll come back to that in a second. Methyl groups, the carbon-hydrogen bonds, we've already talked about those. A really important molecule we see where we see a lot of phosphate groups is ATP. So this is adenosine triphosphate, 3 phosphates. It is the energy currency of the cell. In fact, this is one of those things, top 10 things I would hope you'd remember or already know, but maybe remember from this class 10, 20 years from now, that ATP is the energy currency of the cell. And why is it a great energy currency? We have these, you could think of them as high energy bonds between these phosphate groups. So when we take our triphosphate and we peel off one of these phosphates, here it is, and you end up with ADP, diphosphate. What also gets released is a bunch of energy, and this energy can go into building more ATP, or it can be used to do all sorts of other things, and that's why this is known as an energy currency of the cell. Let's put these functional groups together now into bigger things. And we start to now look at what are our major macromolecules that we find in living things. All right, welcome to my kitchen, a great place to do biology. So our 4 macromolecules that we find in living things are carbohydrates, nucleic acids, proteins, and lipids. Let me ask you a little question here. If I take this chicken breast and I'm going to thaw it out and cook it up for dinner, when I eat this, what macromolecule or macromolecules am I eating when I eat this? So maybe some of you got this right away, or maybe some of you first checked off protein, because when we think of meat, for example, we often think protein, and it's true that meat contains relatively more protein than maybe other things, but it also still does contain those macromolecules because it came from a living thing, and living things are made out of those 4 macromolecules. The same could be said for a potato, right? When you think of a potato, you might think, carbohydrates, lots of carbs in potatoes. True, but there's also those other 4 macromolecules in potatoes. So if you're starting with something that was a living thing, you have to go back to the idea that all of those macromolecules are in there. Now that might not be true for highly processed foods. This chocolate bar here, although it does contain almonds, those almonds are going to have all those macromolecules. The actual chocolate well, once was a plant, at least parts of it, so maybe there's trace amounts in there, but highly processed foods maybe don't have all of those macromolecules in it. But if it came from a living thing relatively recently with minimal processing, then it contains all of those macromolecules. Now let's talk about how those work. So here are our four main macromolecules. As I've already mentioned, one of the patterns that we see with at least 3 of 4 of these is that they form chains of monomers. So these are polymer molecules made up of repeating units. Those units, as we'll see, can sometimes be different, but they form long chains. The exception to this in our 4 groups of macromolecules are lipids. Lipids don't form long chains. One example of lipids are fats. That's not the only kind of lipid, but those do come together in a similar way to the other macromolecules. You'll see how that works, but they don't really form long chains like the others do. So those that do form chains come together in this manner. So what we have here is adding monomers to our growing polymer chain. And this is just a theoretical construct, but this would work for carbohydrates, proteins, nucleic acids. Is this reaction called a dehydration reaction or dehydration synthesis. And what's being dehydrated is your polymer and monomer. So what's happening here is the monomer comes along and gets added to the chain, and water is released. Now it looks like it's producing water, but remember what we're talking about is that actual bigger compound. We're actually taking a water molecule's worth of hydrogen and oxygen out of that polymer. And now this whole thing is 1 unit longer. So this was 3, now there's 4 units. To take these apart, they are broken down by hydrolysis. Hydro means water, lysis means breaking. So hydrolysis is breaking by water. And so what's happening here is water is being put back into the polymer as this 1 unit is being broken off, or maybe this is even longer and the break is happening here between 2 longer pieces. Either way, water needs to be included in this reaction to replace where these, now these chemical bonds are going to be open, the hydrogen and the hydroxyl group here. And so this is what we see with our chain-forming molecules like carbohydrates, nucleic acids, and proteins. So let's talk about those. We'll start with carbohydrates. Carbohydrates, they're made up of carbon, hydrogen, and oxygen. And the basic monomer unit of carbohydrates typically contains 3 to 6 carbons in terms of conventionally what we would consider a carbohydrate. Obviously, you start putting those together in longer chains, that formula, chemical formula gets much more complicated. But I'm just talking about those basic units. So what are examples of carbohydrates? Let me think about that for a second. You've probably come up with a few. You might have said something like glucose or sucrose or fructose. Notice that ose, O-S-E ending. And when you see that, you're usually dealing with a carbohydrate. That doesn't mean every carbohydrate ends with O-S-E, but that can be indicative of carbohydrates. All right, carbohydrates are thought of as good energy storage molecules. So think sort of sugar packed with energy. And we'll see when we get into metabolism how something like glucose is kind of the fundamental starting unit in a lot of metabolism. And why are they good energy storage molecules? Because in those compounds you have really weak bonds or less stable bonds, and moving that by breaking it down into a lower energy state in carbon dioxide and water, you release energy, and that can be used to do all kinds of things. All right, so our examples of carbohydrates, a few examples are here. Glucose is one of the focal carbohydrates for an intro biology class. Glucose is a monosaccharide. So the saccharide refers to the sugar, carbohydrate. Mono means 1, so it's a single sugar. So glucose we see over here. It contains 6 carbons, but note, there's other sugars that contain, like ribose, 5 carbons, or glyceraldehyde, 3 carbons. And where double bonds are, how many carbons there are, like we talked about with isomers, this is going to determine what that actual function of that compound is. Like fructose here is an isomer of glucose, right? So you have the same chemical formula, but you have a double bond in a different place, and then you've got something that's different. These linear structures may be helpful for counting up the carbons, but what's usually really happening in an aqueous solution is these will form, or at least the bigger ones here, will form rings. So here is glucose forming a ring structure. Notice here the carbons are numbered. That'll be important when we get into some of the chemical bonds here in a moment, and when we talk about nucleic acids. So we number the carbons, and it's easy to see on the chain here, but remember when it forms this ring form, you have to keep track of where those carbons are. So 1, 2, 3, 4, 5, 6. This is going to be something that you see, this like a hexagonal ring looking structure, or even where it just shows the ring and not all these other functional groups attached. So that's a monosaccharide. Let's start putting these together. So you put them together by dehydration synthesis. So here we have glucose and glucose. We lose a water here, so this is dehydration, and we form a chemical bond. Notice this bond is specifically called a 1,4 linkage, glycosidic linkage. It's talking, the 1,4 is referring to the carbon here, that's carbon number 1 on this molecule, and carbon number 4 on this molecule. So you can tell something about where those bonds are taking place between two monomers, depending on these numbers here. So now we've created maltose, which is a disaccharide. Di means 2. Here's another one. Glucose and fructose come together, dehydration synthesis. This is a different type of linkage. This is a 1,2 linkage. So carbon number 1 on glucose, carbon 2 on fructose, and you create sucrose, which is sugar, table sugar. And that's that 1,2 linkage. Carbohydrates can be put together in these long chains like we're seeing. We had disaccharides, now we're making trisaccharides, or really polysaccharides, in these long linear chains, but they can also branch off. So here's a 1,4 linkage between these two monomers, but this one also has this 1,6 linkage, and this creates a branching structure like we would see here with glycogen. It doesn't just form one single chain, showing as this in this coil, but you have a branching chains coming off of those individual chains. So what are the functions of these carbohydrates? Well, we can go through them fairly quickly. We can broadly divide carbohydrate function into storage and structural. Now, there's all sorts of little carbohydrates that'll come up a little bit in this class over the semester, where we see them being important on cell surfaces and other interactions, but broadly storage and structure. So storage, energy storage, in plants, we see this happening in starch granules, so amylose and amylopectin. In animal cells, we see this with glycogen. So in your liver stores sugars in this big complex molecule called glycogen. Those individual glucose molecules can be broken off as they're needed, but this is how energy is stored. Structurally, think like a tree, the wood structure of a tree, or even down at in the leaves, the cell walls of the leaves, they contain cellulose amongst other things, but cellulose is a major structural carbohydrate. So in cellulose, you have these long chains of glucose monomers, as we're seeing here. And they're also cross-linked. So you see these little dotted lines here, and it helps provide connections between these long fibers. Think of almost like a cable. It provides that strength, and then you have this structure. In animals, you don't have cellulose, but you do have something called chitin in, say, arthropods, for example. And you see something similar where you have these long chains of carbohydrates. There is an interesting subtle difference here. You have actually a nitrogen group on those carbohydrates. So you don't usually see nitrogenous things in carbohydrates, but you do in chitin. And so these are the kinds of carbohydrates you find in animals. What about fungi? So do fungi have structural and storage carbohydrates like animals or like plants? All right, so you had a couple of choices there, and you end up on the right answer eventually. Animal is the answer. And why is that? Well, this is where we kind of go back to the idea of relationships between all living things. Fungi are more closely related to animals than they are to plants. So we sometimes might look at fungi and sort of think they're kind of like plants, but they are more closely related to animals than plants. And that's why we see some of these basic fundamental similarities with them. Our next group of chain-forming macromolecules is the nucleic acids. So when you think of nucleic acid, hopefully you're jumping right in your mind to DNA. RNA is also a nucleic acid. There's smaller molecules that are nucleic acids too, which we'll touch on briefly. But the main functions of these compounds are to store, transmit, and express or help express hereditary information. So these are the storage molecules for what makes an organism as it is and how that information is passed from one generation to another. A very important concept, which we'll see again, and this is another one of those top 10 things, is this idea, or sometimes this is called the central dogma of molecular biology, this idea that expression of genes goes from DNA as sort of the coding information, the repository, the instructions, to RNA, which is this sort of temporary copy that is used and taken to the factory that builds proteins, the ribosome, and that produces protein. So DNA to RNA to protein. Put it on your top 10 list. And clearly, nucleic acid plays a central role in this all throughout. You'll also see how that ribosome, that protein building machine, it's actually mostly made out of RNA. All right, but for now, what are the sort of basic things structural things that we see with nucleic acids? Well, they're built out of these monomers called nucleotides. So here's an example of a nucleotide, and a nucleotide has sort of three components to it. It has a phosphate group attached. It has a sugar, so a 5-carbon sugar, a pentose, and then you have these nitrogenous bases. And so when we think of like A, C, G, and T, this is what determines whether that's an A, C, G, or T. Other things to pay attention to is this, when we put them together in chains by dehydration synthesis, is you have this repeating sugar, phosphate, sugar, phosphate. This is called the sugar phosphate backbone, and those bases project out to one side of that sugar phosphate backbone. So let's look a little more closely at these bases. So the bases come in two kinds. They're called purines and pyrimidines. And the pyrimidines are C, T, and U, or cytosine, thymine, and uracil. The purines are adenine and guanine. You can come up with all kinds of mnemonics to help you remember this, but one that I had learned a long time ago was purines. Think pure as gold. So purines are adenine and guanine, Ag. And for the pyrimidines, you say pyramids are pointy and might cut you, C-U-T. Again, come up with whatever works to help you remember that. So here's what they look like, a little more detail. And you can see the shape of these, the structure is different. So pyrimidines have this six-sided ring, whereas the purines have an additional ring attached to that six-sided ring. And it's important to note here that in DNA, you have thymine, but not uracil. And then in RNA, you have uracil, but not thymine. Oh, and both DNA and RNA have A's, G's, and C's. The fundamental difference between RNA and DNA is incredibly minuscule, but very important, and it's this sugar. So this pentose sugar, it's ribose, as I already mentioned. In RNA, it's ribose, and there's an OH group here. In DNA, it's deoxyribose. The difference between these two is a single oxygen molecule. So here's a hydrogen here instead of an OH. That's what makes the difference between these two. Of course, the different bases as well, and some of how they behave is going to be different. But fundamentally, at a structural level, it's that sugar that makes them DNA versus RNA. When we're building new strands of DNA or RNA, the new monomers are added only on one end. So they're added on this so-called 3' end. They don't get added to this other end, the 5' end. Why do we call it 5' and 3'? It has to do with those numbered carbons on the sugar. So here's this 3' OH on either DNA or RNA. That's where the next nucleotide gets added. All right, so what are some other things? Well, one is, remember that sugar phosphate backbone I mentioned? It's connected, or those bases are connected through these sugar or phosphodiester bonds. So you got phosphate with those oxygens, and that creates a phosphodiester bond. The strands are polar, meaning, well, polar, they are soluble in water because they have a strong negative charge, but polar in the sense that they have endedness. So they go from 5' to 3'. Let me go back here. It has a 5' end and a 3' end. So they're not symmetrical. And if we put or fold RNA or put two strands of DNA together, they're held together by hydrogen bonds across the nucleotide bases. So here, just reiterating some differences between DNA and RNA. DNA typically is going to be found as being double-stranded. So you have one strand of DNA running one direction, one going the other. In terms of that 5' to 3', they go in opposite directions. They're anti-parallel. That base pairing occurs between the nucleotide bases. RNA is typically single-stranded. You have uracil instead of thymine, but RNA can fold, as we'll see. So here highlights some of the similarities and differences. Sugar phosphate backbone, yes. The sugar deoxyribose versus ribose. The bases are different, or one base is different in each. RNA may fold, but it's usually single-stranded. DNA double-stranded, anti-parallel. Length here matters. DNA is typically forms much longer molecules, millions of bases, versus RNA, maybe thousands of bases, and it's usually short-term stable. Complementary base pairing, that sort of relates back to this strandedness, absolutely. And RNA sometimes if it folds. So let's see what that looks like. So here in A, here's DNA. We've got this very uniform base pairing, these strands running opposite directions, 5' to 3' and 3' to 5'. RNA usually is produced as a single strand, but then might fold up on itself. If the bases match up, like if we zoom in on this one part of the molecule, these bases match up, they will pair with each other. And so these things will fold, and that folding can be super important. So in this case, this is a transfer RNA. As when we see how proteins are built, this is important for bringing amino acids to the ribosome to be added to a growing protein. And so how this thing folds is super important. So it's not just sort of random how it folds. How it folds determines its function. A couple of other really important nucleic acids. Well, ATP, we've talked about this one. Energy currency of the cell, that's a nucleotide. So this is like a monomer, a building block of let's see, is this a building block of DNA or RNA? All right, you had a 50-50 shot, and if you didn't get it, you get it on the second try. But the key here is we see this OH group. Oh, sorry, over here, this OH group. That means we're dealing with RNA. So ATP, energy currency of the cell, that is an RNA nucleotide. So it can be used as energy currency or building another strand of RNA. Another important one, or actually two important ones, although they're very similar in structure, are NAD and FAD. These we'll see are really important electron carriers in metabolism. And look, it looks a little different, but you've got a nitrogenous base, you have sugar, pentose sugar, and nitrogenous base, and phosphates connecting them. So it looks a little different, but this is a kind of nucleic acid. All right, one last topic in part one here is this concept of genomics and proteomics. This is something that you will see as just a common part of your lives going forward. This has become a cornerstone of research, gathering huge sets of data. When I was in college, the idea of sequencing a human genome was just, still a few years away. But from that first genome sequenced, even of a bacterium to a human genome to now this becoming commonplace has happened very, very quickly. So nowadays, a human genome can be sequenced in a few days. Making sense of all those data, that's another huge challenge. And that's the job of bioinformatics. So this is using computer software, data analytics to generate reports and determine what's going on in that genome. So genomics is this study of humongous data sets or full genomes of individual organisms. And another field here is proteomics. What do you think that is? Well, that is just the study of all the proteins in an individual, which, as we'll see later, that's actually a much more complicated task. All right, so that's it for part one. Tune in for part two.